Chapter 3, Topic 3.4, Bacterial Internal Structure. In this topic, we will be discussing the internal structures of prokaryotes, including the contents of the cell cytoplasm, which includes cytoplasm, chromosome and plasmids, ribosomes, inclusion and granules, and the cytoskeleton. We're also going to be talking about bacterial endospores. At the end of this recorded topic lecture, you should be able to meet the following objectives. Identify 5 things that might be contained in bacterial cytoplasm. Name the functions of the 5 things that might be contained in bacterial cytoplasm, and detail the causes and mechanisms of sporogenesis and germination. Let's talk briefly about the cytoplasm. The cytoplasm is a gelatinous or jelly-like substance on the inside of all cells. It's mainly water. Think of it as anything that's inside the cell that's not solid. On to the chromosome and plasmids. As you already know, bacterial DNA is not enclosed in a nucleus. It resides in a region of the cell called the nucleoid. Bacterial DNA is generally in one circular double strand called the chromosome. The chromosome is coiled around special proteins so it will fit into the cell, so it will be protected, and so the cell can regulate what genes are turned on and which are turned off. Some bacteria have other, smaller circles of DNA called plasmids. They generally carry what I call superhero genes, such as for resistance to antibiotics, toxins, enzymes, and so on. So plasmids basically make the bacterium stronger than it was before it got the plasmid. Ribosomes, which are represented by these little dots in the diagram, are non-enveloped organelles that make proteins using the information from the chromosome and the plasmids. They are composed of protein and RNA. They come in 2 parts or subunits. The size of the molecules is expressed in S. This comes from the rate of sedimentation in a centrifuge, since we don't have scales small enough to weigh them. Bacterial ribosomes come in 50S and 30S subunits, which combine to make a 70S ribosome. No, 50 and 30 do not make up 70, but I wouldn't worry about it at this point. Eukaryotic ribosomes are 80S, which is important to remember for later. In bacteria, in good times, when food is plentiful, they can store the extra food in the form of inclusion bodies, these pink guys here. Think of it as bacterial fat deposits, but they aren't always fats or other lipids. Some inclusions are full of gas, so that aquatic bacteria can float up and down in the water column. For example, photosynthetic bacteria can float to the surface during the day and sink during the night to get away from dark-loving predators. Some bacteria store waste products in inclusions until they get rid of them. Some sulfur-producing bacteria do this. Back in the old days, when I was studying this, we thought that bacteria did not have a cytoskeleton similar to what you would see in eukaryotes. We thought that just the peptidoglycan cell wall influenced the shape of bacteria. But as it turns out, at least some of them, mostly rods, have a cytoskeleton similar to what you would see in a eukaryotic cell. I don't have a diagram of this in a prokaryote because it's so new, but they do have actin filaments like eukaryotic cells do, and they also have microtubulin, which is similar to eukaryotic cells. Currently, we think that the actin and the microtubules influence how the peptidoglycan gets laid down in the cell wall, thus influencing cell shape. On to endospores. We're going to discuss their function, formation, and germination. Endospores help bacteria survive extreme conditions, such as extremes of heat, radiation, drying, lack of food, and so on. They are one of the most resistant forms of life. Bacteria that produce endospores have a 2-stage life cycle. The vegetative phase, or stage, is the growing, eating, and reproducing stage. When times get tough, such as if the environment starts to dry out or if food runs low, the vegetative cell will form an endospore. The endospore is the resting stage with very low levels of metabolism. That's part of what makes it so resistant to everything. It's sort of in suspended animation for you sci-fi aficionados out there. Endospores are not reproductive. One vegetative cell makes one endospore, which makes one vegetative bacterium. To be reproductive, you have to have more cells afterwards than what you started out with. Here's a lovely diagram from the book showing the formation of endospores and their germination. The main thing that I want you to get from this is one endospore forms one vegetative cell. After endospore formation is complete, the vegetative cell's DNA is degraded. And the mature endospore is wrapped in multiple protective layers. Now, for germination, the main thing I want you to remember about germination is one endospore forms one vegetative spore. Think that's going to be on an exam? Now, let's talk about what can cause endospore germination. Water can cause it. That makes sense, since drying can stimulate endospore formation. The presence of food can cause it. Once again, makes sense. The important thing to keep in mind is that a short period of boiling can also cause germination. Many is the home canner who has unwittingly stimulated botulism endospore germination by not getting the pressure cooker temperature high enough. If you can at home, make sure you follow the directions and get your equipment checked to make sure it's functioning properly, or you could cause a group funeral. Unfortunately, this has happened in the past. On to medical significance. Most spore-forming bacteria are harmless soil bacteria that are just trying to make a living in a harsh world. But the genus Clostridium, which is composed of soil bacteria, has several pathogens. One I've mentioned, Clostridium botulinum, which is the cause of botulism. There's C. tetani. Guess what that causes? And C. perfringens, which is the cause of gas gangrene. Lately there has been a real problem in hospitals and care centers with C. difficile, which can cause severe diarrhea and a serious type of colitis. I will be mentioning our friend C. diff again. Another medical significance is because endospores are so hard to kill, they are used as the standard for testing sterilization systems. But more about that later. Well, that's it for this topic. You should now be able to identify 5 things that might be contained in bacterial cytoplasm, name the functions of the 5 things that might be contained in bacterial cytoplasm, and detail the causes and mechanisms of sporogenesis and germination.